Well, welcome everyone. I know some of you have joined us for past Scientist Hours. This is our monthly Scientist Hour, and we are going to be featuring our inherited eye diseases. My name is Sarah Kelly, and I'm the Senior Development Manager at the Retina Foundation. Thank you for being with us. The mission of the Retina Foundation is to prevent vision loss and restore sight through innovative research and treatment. Each year, we have the opportunity to see 2,300 patients completely free of charge. And they're typically in our three core areas, which are pediatric eye conditions, inherited eye disease, and age-related macular degeneration. Vision loss due to inherited eye could start at any stage of life from infancy to adulthood and can affect multiple generations of families. At the Retina Foundation, we partner with people around the country and globe to research the causes, the treatments, and then cures for inherited eye disease. Today, we have Kaylie Webb, who is going to be presenting on what we do with genetic testing at the foundation, and we're really excited to share more about what we do. The conversation series for the Scientist Hour was to share the most up-to-date research from our scientists at the Retina Foundation, and we don't want to be the best kept secret. So please, we will be putting the recording on our website, and feel free to share the information with anyone who you think would be interested. We really appreciate that. During the presentation, Kaylie will be taking pauses throughout the conversation and opening up for questions. We really, truly want you to take advantage of that opportunity and take yourself off mute and ask any questions that you may have. If you have any questions outside of those pauses, feel free to use the chat feature in Zoom. And now I would like to introduce Dr. David Birch. He is our scientific director and the director of the Rose Silverthorne Retinal Degenerations Lab at the Retina Foundation. Dr. Birch will now introduce Kaylie Webb, who will lead the discussion.
Thank you so much for the introduction. And good morning, everybody. Glad to have you all on board. So you're in for a real treat today. Kaylie's been with us for many years. And when she began, I think we had maybe 3 genes that we were able to identify. And each test, each time we sequenced a gene, cost many thousands of dollars. And in fact, it was only through the help of the National Eye Institute and the Foundation Fighting Blindness, other donors, that we were able to do this at no cost to the patient. So we very, very early on were testing patients for the known genes, which in that case was only a few. And again, it cost thousands of dollars per gene. So over the course of Kaylie's time here, the cost of testing has dropped fantastic, dramatically. And we can now do rapid sequencing. We can parallel sequence many, many, many genes at one time. And in fact, our current panel has about 280 genes on it that we're screening. So we went from a situation where just a tiny fraction of the patients with retinitis pigmentosa and other allied diseases identified to a situation now where we can identify it in about 80% of patients. So this has really dramatically changed our role and the way we do research, and has led to a lot of the clinical trials that we're participating in. So Kaylie's been on board, the key person in the Southwest Eye Registry and the genetics, and she's going to share her knowledge and experience and give you a chance to ask questions. So please do ask questions at the breaks. So without further ado, here's Kaylie Jones.
All right, well, thank you for the nice introduction. I'm gonna just share my slides real quick and get the presentation started. Feel free to ask questions at any time. I just want to, thank everybody for taking time out of their day to join us this presentation. So today I'm just gonna, kind of start off by discussing a little bit about what inherited retinal dystrophies are and what causes them. Then I'll talk a little bit about the genetic testing process that we have here at the Retina Foundation, and then show a little bit about the complexity of retinal disease genetics as well. And then I'll kind of conclude by talking about some of the genotyping or genetic testing efforts that we've been involved with, over the years, and what we do with that genetic testing information and how we use that for clinical trials. So to start off, inherited retinal dystrophies include diseases such as retinitis pigmentosa, commonly referred to as RP, choroideremia, Stargardt disease, cone and cone rod dystrophies, and other syndromic forms of retinal dystrophies such as Usher syndrome, which oftentimes includes Usher syndrome is RP and patients who have some degree of hearing loss. These are all rare retinal diseases that lead to vision loss and blindness. So to kind of get you guys involved, from the beginning, we have a poll here for you that we're going to pop up. And we're going to ask how many people are affected with these types of eye diseases. We're really asking what the prevalence of these diseases are. And your options are 1 in 3,500, 1 in 10,000, 1 in 72,000. Just give you guys a couple of seconds to answer there. Just give you guys a couple more seconds. I think we have about 62% of people who have voted. And I'm going to go ahead and end the polling, and we have the results here. I think you guys can see those. About 48% of people chose 1 in 3,500, which is the correct answer. About the other half of participants selected 1 in 10,000. So you guys were spot on there. Retinal dystrophies are characterized by degeneration of different cell types within the retina. The retina is a thin piece of tissue lining the back of the eye. This tissue converts light into electrical signals, which are then interpreted by the brain as, vision as sight. Retinal cell types affected by retinal dystrophies include the rod photoreceptors, which are distributed throughout the retina. These cell types are primarily responsible for vision at low light levels. And then there's also cone photoreceptors, and these are concentrated within the fovea, the central area of the retina. And these cell types are primarily responsible for color vision and your central visual acuity. So the timing, progression, and range of symptoms associated with retinal dystrophies can really differ from person to person. And a lot of times those symptoms depend on what types of photoreceptor is predominantly or initially affected with the disease. Some of the symptoms of retinal dystrophies can include the following. Patients can have impaired dark adaptation where they have night blindness. They can have loss of peripheral vision, also known as tunnel vision. Patients can also experience reduced central vision or impaired color vision. Oftentimes patients will experience, 1 or more of these symptoms throughout the course of the disease as well. A common other symptom that patients experience oftentimes is photophobia, where they have sensitivity to light, bright lights, or they may experience flashing or blinking light sometimes. So retinal dystrophies are usually caused by 1 or more mutations in a gene. There's currently over 300 different genes known to be associated with retinal dystrophies. And Dr. Birch kind of mentioned this, when I first started working here at the Retina Foundation, we would have a patient come in and we would test their DNA sample for a single gene and hope that we had the right one that we were looking at. And then we would kind of go sequentially down, maybe 2 or 3 other genes that might possibly be the cause of their retinal disease. And so things have really changed to where we've identified way more genes than we ever imagined existed for causing retinal diseases. And we're able to test a lot of these genes, in panel-based tests where we test them all at one time. And I'll talk a little bit more about that process in just a few minutes. And so, like I said, we use DNA sequence analysis to identify, the exact genetic cause of each of our patients' retinal disease. And the detection rates have gotten pretty good using the based test. So right now, detection rates are currently at approximately 75%, meaning that for roughly a quarter of all of our IRD patients, we still can't identify the genetic cause for them. So there's still some work to be done. And part of our research efforts here at the Retina Foundation, is to work on discovering those new genes that might be causing some of these unsolved cases, or maybe to learn a little bit more about different genetic variants within already identified genes that maybe we can't detect yet with our technology. So before I start talking a little bit about each step in the genetic testing process, I'd like to kind of take a brief moment and see if anybody has any questions that they would like to ask.
We do have one in the chat. It says, is sequencing done on site at the...
So it used to be done on site. We would do a limited amount of sequencing on site, but now most of it is sent off to either a collaborating research laboratory or a diagnostic laboratory that will do the testing. So it's just a lot cheaper just to do it off site than it is, to do it in-house. Are there any other questions? All right, so now I'll just kind of...
I just received one as well.
Okay.
Is there a certain geographic area that is specific to where the genes are found, or is it worldwide pretty diverse?
It's worldwide. These retinal dystrophy genes have been found in, all types of populations across the world. They're not specific to a certain ethnic group. But that's a good question. There are a few of the genes that have been, identified in certain regions, initially, but then as we start screening larger populations, we've started finding those same genetic variants, scattered in other populations as well.
If I could just jump in for 1 second, that's an interesting conversation point.
It is.
Because there are new genes there are new mutations that pop up. I mean, they're what we call de novo cases. And often when you have a new mutation that pops up, it sort of is more focused in one part of the world or one part of the country. And it kind of gradually, that mutation spreads around the world eventually, just over dozens of years. Most of the mutations we're looking at, though, have been around for a long time. But there are a couple that are specific to the United States, and there's one that's almost unique to Finland. And so, there are a few examples like that.
Yeah, there's one that us and another research group, did some work on where we identified a dominant RP gene that seemed to be in Hispanic populations that were isolated to kind of the southwestern US. And so that was kind of interesting. It was about 5 or 6 years ago we identified this gene. It's called the SAG or SAG gene that causes dominant RP. But recently they've added this gene to a lot of the retinal dystrophy panels, and I've started seeing it in other patients, who don't have a Hispanic background, or maybe they did several generations ago, but they aren't aware of it. So it's just kind of interesting. And we found another gene that was another dominant RP gene that was prevalent in French Acadian populations, specifically patients from Southern Louisiana who had French ancestors. So that one was pretty interesting as well. Any other questions for me?
We have 2 others in our chat. It says, can you be sensitive to bright lights, especially at night, and not have a retinal problem?
Well, I think there's some other issues that can cause, like glare specifically at night. I know a lot of people have issues with driving at night or in the rain and have trouble with, street lights and glare. And that might not necessarily be because of a retinal disease, like an inherited retinal dystrophy. It could be something else like myopia or other issues.
And then the last one we have right now is, are these mutations hereditary?
Yeah, these mutations are hereditary. And I do have some slides where I'll kind of show the different inheritance patterns for some of these genes. And maybe that'll answer that question a little bit more in just a couple slides. Okay, so I'll kind of move on and just talk, briefly about the genetic testing process at the Retina Foundation and kind of how it works. So typically what happens is a patient will be diagnosed, or they'll be suspected to have a retinal dystrophy. Maybe they've been diagnosed with retinal dystrophy many years ago, and now they're just kind of starting to get interested in participating in research. And they'll oftentimes, get referred to the Rose Silverthorne Retinal Degenerations Lab run by Dr. Birch here at the Retina Foundation. So patients will typically come in where they'll get a comprehensive visual function exam. This will include visual fields testing, electroretinograms, ERGs, and various types of retinal imaging. And then at these appointments, what we typically do is we start the genetic testing process for the patient. So here's just a schematic of what happens. Typically, we'll sit down and have a discussion of their personal and family history. A lot of times we'll ask their health-related questions because a lot of times our panel-based testing does include syndromic forms of retinal disease. So we make sure we get a good medical history as well. And then we will talk about different genetic testing options, whether a patient wants to participate in a no-cost study-related testing, or if maybe they don't want to do that, if they want to, do a fee-for-service testing, we'll talk about those options as well. And then, we just kind of take everybody's, case on a case-by-case basis and see what the best route for obtaining that genetic testing is. So once we decide what we're going to order and, what the expectations are from the testing, we'll collect a DNA sample. Usually we'll either collect blood, buccal swab, which is just a cheek swab, and we send that out to the lab for analysis. And so this analysis typically involves next generation sequencing of known retinal dystrophy genes. And the panel we are currently using is a 322 gene retinal dystrophy panel that includes 285 genomic genes and then 37 mitochondrial genes as well. Then once the results are ready, it usually takes about 6 to 8 weeks to get those results back. They get sent back to us and we review their genetic results along with their clinical evaluation to kind of get the full picture of what's going on for each patient. And then finally, the results get discussed with the patient. We make sure that they understand the implications of the findings and what the risk to other family members or their children or for future children might be from those genetic findings. So there's several reasons why genetic testing is highly recommended for patients who have retinal dystrophy. First of all, the use of diagnostic genetic testing can be used to, confirm a clinical diagnosis or maybe to differentiate between two forms of retinal dystrophies. For example, sometimes you might have a young patient coming in who's experiencing night vision issues, and we may want to determine if the patient has, a progressive form of retinal disease such as RP, or maybe a more stationary form of night blindness called congenital stationary night blindness. And so the vision testing along with the genetic testing can oftentimes help us differentiate between those two diagnoses for the patient. Another part of genetic testing is the counseling, and this is where the results get interpreted in an understandable manner. And again, we talk about the recurrence risk with the affected family member. Oftentimes, the genetic testing helps identify what the inheritance pattern is for each patient and what the odds of passing that gene mutation onto their children are. And we can use an identified gene mutation to test other families who maybe aren't yet symptomatic or who might be at risk for inheriting that gene mutation. Additionally, an increasingly more important reason that all the retinal dystrophy patients should undergo genetic testing is to see if they might be eligible to participate in a clinical trial. So as more and more advancements are made in developing potential therapies, it's really important to know what specific gene mutation is causing each patient's retinal dystrophy. That way we can be aware of clinical trials or advancements that are specific to their form of retinal dystrophy. All right, so here's where I have some slides about the different inheritance patterns that go along with these retinal dystrophies. So there's been hundreds of different genes that have been identified that can cause retinal dystrophy, but understanding how these genes can impact vision can be really complicated in some cases. For one thing, the inheritance patterns, the way the gene mutation is passed from generation to generation isn't always straightforward. There's 300 different genes, but they all aren't inherited in the same way. So one of the ways that they can be inherited is what's called an autosomal dominant pattern, meaning that you only need one mutated gene to be affected with the disease. So here we have a 4-generation family. The squares on this pedigree represent males, the circles represent females, and the shaded in symbols show an individual who's affected with a retinal disease. So for every affected individual in this family, each of their children would have a 50% chance of inheriting the mutation. And so you can see here, there's quite a few individuals in this family who have been affected with the retinal disease. So autosomal dominant inheritance, isn't always straightforward as it sounds. With dominantly inherited genes, some of them in particular, like the PRPF31 gene, PRPF3 gene, those can result in something called incomplete penetrance. Again, this is the same family as before. But right here, you have an individual who, had a parent who had a retinal disease, and then they ended up having children who had retinal disease, even though they didn't have any signs or symptoms of the disease. And so in this case right here in this family, this individual inherited the genetic variant, but never expressed symptoms, but was still able to pass it on to their children. And so it's really important to understand the specific gene that you're looking at for each family and to know if that's a possibility as well. Another form of inheritance is autosomal recessive inheritance. This is where an affected individual inherited 2 mutated copies of a gene, one from each parent. So in this family, you have 4 siblings, and 2 of them have the retinal disease and 2 do not. And for this family, both of the parents of the affected individuals were carriers of the disease gene and happened to pass both faulty copies of the gene onto the affected children. The other form of inheritance for retinal dystrophies is X-linked inheritance. This is where the retinal disease gene or the mutated gene lies on the X chromosome. So males have an X and Y chromosome and females have 2 X chromosomes. And so typically the disease for X-linked traits really only shows symptoms in males, but not with all retinal disease genes. In some cases, some of them, the females can have some degree of symptoms as well. And they can kind of almost look like a dominant inherited family from the get-go. So these types of inheritance patterns are associated with genes that are found in genomic DNA, which is found in a cell's nucleus. But there also are some retinal disease genes that are located in the mitochondria. And these types of genes don't really follow traditional inheritance patterns. So with mitochondrial related retinal diseases, patients can either be male or female and be affected, but within a family, they're gonna all of the affected individuals will have the same female relative, since mitochondrial genes are only inherited from the mother. So, they can be quite complicated here on the inheritance patterns. Since we had some questions earlier about inheritance of retinal disease genes, I think it'd be a good point to stop and see if anybody had any questions about inheritance patterns.
There is one question in the chat. It says, when tests have a specific gene mutation, how accurate is the result?
So most of the testing will have, accuracy statistics published in it, but most of the time it's over 99.9% accurate. I think our retinal dystrophy panel has an accuracy rate of 99.98%. So they're quite, and a lot of the laboratories do have, quality checks in place to make sure that they can verify the mutation before they report out the result.
I have another one here. It says, which of these various ways are most common?
That's a hard one to answer. They're all quite common. Recessive inheritance is usually the case when you have an individual come in who has no family history. That's usually what we would start with assuming, is that it would be a recessively inherited gene if it's just, an individual who shows up with a retinal disease. That's why it's really important to get a good family history whenever we have somebody come in and to, see how many generations are affected. And that can really help us to pinpoint what type of inheritance we're looking at. Any other questions? All right. So I'll go on to my next slide here. So going forward, I'm going to talk a little bit about variable expressivity of inherited retinal disease genes. So what we're talking about here really is phenotypic variability. A lot of these retinal disease genes are highly heterogeneous, meaning that mutations need to be really interpreted in context of each patient's clinical presentation. And so, like I said previously, our retinal degenerations lab, offers both no-cost visual function testing and genetic testing to patients, which really allows us to get a complete picture of each person's case. So variants in a single gene may be associated with different types of retinal dystrophy. So here we're talking about one gene in particular called the PRPH2 gene. And I'm just going to show some examples here of what's called an OCT. An OCT is a device that we use to image retinas, and it takes cross-sectional pictures of a retina so that we can look at the different layers within the retina. So in this first case here, I'm going to show is a 29-year-old patient who has a mutation in a dominantly inherited gene called PRPH2. This gene's been well studied. It was one of the earlier genes that was identified. And this patient has a classic presentation of retinitis pigmentosa. In this patient, she's lost her periphery, her peripheral vision. She has good central vision. She would have trouble with night vision. Here we have another patient who also has a PRPH2 mutation, the 43-year-old female who presents with a totally different phenotype. Here, her central vision would be affected. This patient presents with almost like a vitelliform retinal dystrophy. You can see here that her retinal presentation looks totally different, even though she has a mutation in the same gene that's actually located quite near this other patient who has RP. Here's a 44-year-old female who has almost like a Stargardt-like presentation. As you can see, she's got these almost fleck-like appearance here that looks totally different than a RP patient that we would see. And again, she has a PRPH2 gene mutation. And again, these patients all have point mutations very close to each other within the PRPH2 gene. And then finally here is another PRPH2 patient. It's a little bit older of a patient, but they have a more severe widespread visual impairment where both the cones and the rods are affected. And this patient has a form of cone rod dystrophy. So these are just a few examples of how a single retinal dystrophy gene can be associated with highly variable clinical impacts. And it really shows the importance of having a comprehensive clinical evaluation to go with the genetic testing. So I just showed an example of how different disease mutations in a single gene can cause, very different symptoms. But on the other hand, mutations in multiple different genes can cause the same phenotype or signs and symptoms. For example, talking again about autosomal dominantly inherited RP, there's currently over 30 different genes that have been associated with that diagnosis. And most of these genes can cause very similar signs and symptoms. And the only way to determine the responsible gene is through genetic testing. So over the past few decades, there's been a tremendous amount of work done at the Retina Foundation. Dr. Birch kind of alluded to some of this. We've been trying to identify the genetic cause for each of our patients' retinal disease and to try to help them and their families understand those findings. Our core initiative for this genotyping is called the Southwest Eye Registry, or the SER. What it is it's a regional database of patients who have inherited retinal dystrophies and allied disorders. It was established in 1995. It was created to serve as a formal mechanism to maintain clinical, functional, and molecular genetic data. The SER includes a DNA repository. You can see there, it's a DNA repository of samples from over 9,000 patients and their family members. And those family members include both affected and unaffected individuals. So the DNA samples and the clinical data maintained in the SER have been used for mutation identification and gene discovery research collaborations with other researchers worldwide. We've worked extensively with several other research groups within Texas, like UT Health, UT Southwestern, and Baylor College of Medicine. But we've also had international collaborators over the years, such as in Canada and Germany. So, it's nice to have a great network of other researchers who share the same goals. Another research initiative that the Retina Foundation has been involved in is the National Ophthalmic Disease Genotyping and Phenotyping Network. for short, it's called iGene, a lot easier to say. This is a genomic medicine initiative created by the National Eye Institute in partnership with clinics and labs across the vision research community. And the core mission of iGene was to really facilitate research into the causes and mechanisms of rare inherited eye diseases, and then to accelerate pathways to treatment. So the iGene network actually is no longer enrolling new patients, but they currently have a lot of patients who we haven't been able to identify a genetic cause for their retinal disease. And so we continue to work with them to identify new genetic causes of disease for each of those patients who haven't had a gene mutation identified yet. And that project has led to a lot of new collaborations. For example, we just had a project with Wayne State University in Detroit. And so that was nice to start a new collaboration through that route. Then our most recent genetic testing collaboration has been with the Foundation Fighting Blindness. It's called the My Retina Tracker Genetic Testing Study. This began in 2017, and this program provides for eligible patients to have access to a national patient registry and no-cost genetic testing and counseling. And so since 2017, we've been able to enroll over 700 patients in that program alone. So now I'll kind of move into, what we do with all this information. We have all this vision testing data. We have all this genetic data on patients. We have eager patients who are, excited to participate in studies. What do we do with that information? So right now there's only one FDA approved treatment for just one specific genetic form of RP. That's the Spark product Luxturna. But there's over 322 genetic forms of retinal dystrophy. And so the Retinal Degenerations Lab at the Retina Foundation is currently conducting multiple clinical trials attempting to find treatments for other genetic forms of retinal dystrophy. So we use that patient registry to recruit patients for these clinical trials. And a lot of the clinical trials that are starting right now are really gene specific, meaning that they're looking for specific genetic forms of disease so that patients, they really do need to have their gene mutation identified before they can participate in quite a few of those studies. There's different types of studies that we have going on. The first category of studies that I'm going to talk about are natural history studies. These are really non-interventional studies where we just follow patients who have specific genetic forms of retinal dystrophy, and we just watch how the disease progresses. A lot of times these are done in hopes of a treatment trial starting up in the future. And they really offer invaluable information on how the disease impacts patients. And it really provides good baseline progression data for future studies. We currently have natural history studies for patients with mutations in these genes, the RPGR, CHM, USH2A, UIS. RPGR is an X-linked RP gene. CHM is associated with choroideremia. USH2A causes Usher syndrome type 2A. And UIS is a recessive RP gene. So we also have several gene therapy trials underway for patients affected with different forms of genetic eye diseases. One of them is an X-linked RP clinical trial caused by mutations in RPGR, a choroideremia gene therapy trial, one for Usher syndrome type 2A, and then one for autosomal dominant RP caused by a single specific mutation in the rhodopsin gene. Additionally, we have pharmacological trials underway that are using various investigational drugs, trying to evaluate their ability to slow or stop progression of diseases, such as Stargardt disease, which is caused by mutations in ABCA4. So just to kind of wrap up, I'll kind of focus briefly on one of the gene therapy studies going on at the Retina Foundation. This is a gene replacement trial that's sponsored by AGTC. It's a phase 2 expansion study looking at the safety and efficacy of a gene therapy for male patients diagnosed with X-linked RP caused by mutations in the RPGR gene. So currently there's no approved treatments for this condition. X-linked RP causes difficulty seeing at night and progressive constriction of the visual field. It affects primarily boys and young men. A lot of times it's symptoms pop up in early childhood or maybe early teen years. And it does usually to legal blindness in the mid-40s. So this study uses a type of gene replacement therapy or an experimental vector carrying a functional or working copy of that RPGR gene is given through a subretinal injection in an attempt to, to replace the mutated or damaged RPGR gene. And this clinical trial is being done to demonstrate whether the study treatment will stop progression of vision loss or maybe even help participants see better. And so this is a pretty exciting study that we've got going on, hoping to put a stop to some of this vision loss in some of these patients. So it's been really exciting, to see decades of research into the causes and the mechanisms behind retinal dystrophies translate into potential treatments for patients. I think these clinical trials are really giving a lot of hope to patients. I think everybody who works at the Retina Foundation is really excited to see, things translate from, bench work to actually, implementing things into patients. So here's some of the members of the Retinal Degenerations Lab. None of this work would be possible without them. We have Dr. Birch, who's our scientific director. We have Kirsten, who's our clinical trials manager. Martin Klein, our lab manager, and Tian Liu, our research assistant. So does anybody have any questions? I'd really like to thank everybody for taking the time to join us to discuss a little bit about genetic testing and some of the work we do in the Inherited Eye Disease Lab. If anybody has any questions, I'd be happy to answer those.
Well, we don't know what.
Hi, Vanessa. Vanessa? Can you hear me?
Hi, yes, Robin, I can.
Oh, okay. I meant to say Kaylee, sorry. I don't know why. Okay. I missed half of this, I'm sorry. But the reason I wanted to sign up was that my grandson was just diagnosed with LCA.
Mm-hmm.
And I want to know, I missed part of it and I feel really bad. Is there a way for me to see the full thing again?
Yeah, I think they're going to record it and maybe they can send you a link to it.
Okay. Did you mention anything about LCA?
I didn't really specifically talk about LCA, but if you have any questions about it specifically, maybe we can visit on the phone later and talk about your grandson's case.
Okay. All right. Thank you.
Mm-hmm.
Okay.
There was a question that came in. It says, where is the Southwest Eye Registry located?
Yep. So the Southwest Eye Registry is actually in the Retina Foundation. It's kind of an abstract thing to think about. It's a database of patients. So all of the vision results from the vision evaluations that we do at the Retina Foundation, it all gets put into that database. And then it also is comprised of those freezers. We have a DNA repository. So we have multiple freezers full of DNA and banked cells from patients that we've seen over the years. It's really nice to have that available because we have those samples on hand to be able to, do studies on very quickly without having to ask patients to come in for additional sample collection.
I had a quick question. This is Rebecca.
Hi, Rebecca.
Hey, how are you? Great presentation. I guess my question is, if you had your genetic testing done through Retina Foundation, what that's been 15 years ago, there's no reason to have it done again as long as they were able to identify the gene.
Right, correct, yeah.
And the other question is, do you guys see patients, X-linked patients with the RPGR mutation that are female with significant vision loss? Is that just an anomaly?
No, it's not. It's not an entirely anomaly. Actually, we've had several publications recently, and there's been other researchers who have published lately on females who have RPGR mutations. And it showed that about half of them do have moderate to severe vision loss, and quite a few of them actually just as severe as a male. And so there's been a little bit of work done trying to find out why those females, or some females, have severe retinal disease and some don't. And then that's kind of an ongoing effort to try to find that answer.
Where could I look to see those papers?
I can email you some of them.
That would be great.
I'll just send you some PDFs of them.
Thank you.
Anybody else?
In the chat, we have Prem Patel put gene therapy. And Prem, if you want to take yourself off mute and just share a little bit more about what you're looking to ask, that would be helpful.
Sorry, that was a Google search I accidentally put in the chat, but I was just wondering, I guess, which of the dystrophies that you mentioned does gene therapy look to be the most promising against?
So the one form of gene therapy that's been approved by the FDA is for LCA, a specific form of LCA. And then some of the other ones that are kind of farther along in clinical trials are the X-linked retinal diseases like choroideremia or X-linked RP. Those seem to be the ones that are a little bit farther along, in clinical trials.
Gotcha. Okay, thank you.
Mm-hmm.
I can just say that, we had an exciting day yesterday because ProQR, which is the sponsor of the Usher 2A trial that we're doing, released the interim analysis of the data from the trial. And it looks like it's really been quite effective, the treatment in actually improving the acuity in advanced patients, and certainly in improving the visual field in less advanced patients. So I think that's another therapy that's going to move pretty quickly into the, what's called phase 2-3, and hopefully for approval.
I have a question. Just to talk about, you said that the vision is showing to be improving in some patients for the gene therapy. What is it about the gene therapy that makes it improve? Is it doing something to the cells in the retina, or?
David, do you want to answer that one?
I could. Yeah, I mean, these genes are replacing a missing gene. So in the case of males with X-linked RP, the RPGR mutation means that they're not producing the protein that's needed by the cells, and that protein is crucial in sort of controlling how things are exported and transported within the photoreceptor. So putting a good copy of the gene in provides the RPGR to the cells, which then can improve the trafficking, improve the structure of the cell. So, we kind of go into these trials hoping that we can impact the natural history, slow down the disease. But in fact, when you make cells healthier, you actually get improvements in many cases. And so we've been finding in some of these trials that there are actual improvements that come to the patient rather than just stabilization. So that's exciting.
Well, I think that may be it. What we will do afterwards is this session has been recorded, and so we can send you the information and the link to this presentation in case you have any other questions that come up or would like to review it again. So thank you, Kaylie. Thank you, Dr. Birch. We really appreciate your expertise. It's wonderful to hear all the work that you're doing. We do have 2 other events that I'd love to share with you. We have our Keeping an Eye on Innovation lecture coming up on April 21st, which is a Wednesday, and it's in the evening. We have Dr. Ryvan Geimer, who's going to be presenting. She'll be presenting on age-related macular degeneration. And then our next event after that will be on June 3rd. So typically we have an annual fundraising luncheon, but this year we have gone virtual and we have moved it to the evening, and we have a very special guest. Dr. Anthony Fauci will be speaking, and he'll be talking about science and collaborations and all things COVID. So we hope you may be interested in joining us for that. And if you have any other questions, we'll be following up with all of you. So we'll be happy to answer anything else that may come up in the next week or two. So really appreciate it. And we hope to see you at one of our future events. Thank you.
Thank you. Thanks, everybody.